have the CDK enzyme forming our cyclin complex. CDK stands for cyclin-dependent kinase. Okay, so again, the role of the kinase is through the phosphorylation and thus the control of several signaling pathways, as we've seen in Chapter 9 when we talked about signaling and the control of signaling through the use of phosphorylation. So here is another prime example of the use of phosphorylation controlling another basic signaling pathway, in this case the essential proliferation of the cell, control of the actual cell cycle itself. So it's called CDK, cyclin-dependent kinase, because it only functions in the presence of cyclin. Okay, without cyclin bound in this complex, the CDK does not function. So the way that it works is, in its activated form, you have this phosphate here actually bound. Okay, in its inactive form, you have the phosphate up there bound. Okay, so with this differential phosphorylation at different points of the CDK enzyme, it allows for the activation here, the activation, or the inactivation of the actual processes. And so if you look at figure 1021 over here, we actually see the use of these CDKs, CDK2 specifically first here, during the restriction checkpoint. It is inactive, okay, at this point. So this is when we have the red one bound, the red bound phosphate group. Oh, can't see that. The red bound phosphate group up here, that's the phosphate group that's attached in the inactive form, while down here, the green bonded phosphate group is when we have the active form. So when CDK2 is inactive, that is when we actually have our G1 phase. Okay, the majority of our G1 phase before the actual restriction point. It only passes the restriction point once we actually activate CDK. Okay, in this case specifically CDK2. So we move on with an activated CDK2. But in the G2 phase, we cannot pass the G2 checkpoint, G2M checkpoint, until we activate CDK1, cyclin-dependent kinase 1. So in its inactive form, again, the same basic concept as far as the differential phosphorylation at different points. At one point, you have the inactive form of CDK1, at another, you have the active form of CDK1. Once you have the differential phosphorylation occurring, it allows us to pass that particular checkpoint. And from there, we move on to the spindle fiber checkpoint, where we're using the APC complex. APC, which stands for the anaphase promoting complex. APC, anaphase promoting complex. And so here we have the inactive form of APC versus the active form of APC, and it's just the same basic concept as far as the differential phosphorylation at different places of the actual complex. In one place you have the inactive form of the complex. When you phosphorylate the complex at another place, that's when you activate the APC complex. Same concept as the CDK2 and CDK1. So at this point, using the APC complex, it promotes the actual functions that occur during the anaphase stage of the cell cycle. Okay, and now in the next video, we'll talk about the last concept in this chapter, and that is the concept of cancer, oncogenes, and tumor